So today's Do Now question says, gram-positive bacteria have a thicker cell wall on the outside of their cell. Do you think this would make them more or less protected from antibiotics, and why? So just like one might assume, right, if I have a thicker outer covering, then it seems to make sense, right, that would make me more protected. But here's something we want to think about. What type of bacteria was Addie infected with? Gram-positive or gram-negative bacteria? My nose is so itchy under my mask. What type of bacteria was Addie infected with? Gram-positive or gram-negative?
Gram-positive.
Oh my God, my nose. So it's actually gram-negative. She was infected with gram-negative bacteria, and we know that her bacteria inside of her were extremely resistant to antibiotics. But if gram-positive have the thick cell wall, one can kind of assume or infer that gram-negative then would have a thin cell wall. So now we have to think about, so it's not the thickness of the cell wall that would make a bacteria more or less resistant to antibiotics. So then what is it? Okay, what is it exactly? So that's what we're building up to find out in just a couple lessons. Oh my God. You guys ever get a fuzzy inside your mask? Ugh. Ugh. Relief. Okay. Sorry about that. So now we're going to go over your homework from last week, which was your 3.35 types of bacteria and antibiotics. So if you want to follow along, you can go to your Google Drive, search for the title, and open it up. Again, we are going to go over your 3.35 types of bacteria and antibiotics homework assignment. Okay, the very first part of this reading was on just types of bacteria and their structures. So we're going to go over just those questions first. Okay, number one here says, what are bacteria? Who could answer that for us? What are bacteria?
Single cell organisms.
Oh, they're single-celled organisms, meaning they're made of just one tiny cell, and that one cell completes all necessary living functions. Good. Number two, what are the three shapes bacteria can come in?
Viral.
So one spiral, yeah, what's another shape?
Rod shape.
Rod shape, good. And
I don't know how to say the other one. Spherical or something.
spherical, or I could just kind of circular shaped, good. So number three, what is the fancy science term we use to describe bacteria that are shaped like spheres or circles? You can type it in the chat, spell it for me. I guess whatever you're comfortable with. I know it might feel a little weird to say it.
Spherical.
Okay, so what's the fancy science term, though, to describe bacteria that are shaped like that?
Circular.
Nope. But it does begin with a C. Came from the reading.
Is it like Coke? It's like.
Yes, C-O-C-I.
Okay.
So you say that coci. You may have heard this before if you've ever heard in any of our videos or readings, or maybe at a doctor's office, they often describe staph bacteria as Staphylococcus, C-O-C-C-U-S. And that ending coccus part actually comes by saying the type of bacteria that have infected you, like when you have strep throat, it is streptococcus, right? It's a strep species of bacteria that's shaped like circles. Okay, so that second part of the name in bacteria tells us what its shape is. Number 4, what term do we use to describe bacteria that are shaped like rods?
Don't it start with a B?
Yes, it does.
I don't know how to say it.
God, there's a fuzzy right here in my mask. I can feel it tickling my nose.
What Kendra just said in the chat.
Okay, Kendria is correct. Bacilli, or we could say bacillus. So a lot of bacteria that you see in yogurt or probiotics that are really good for our digestive system are bacillus bacteria. And again, if you see bacillus, it tells us that it's rod shaped, which means it's kind of shaped like a little tic tac. And then it tells you the type of species of bacteria it is. Number 5, what term do we use to describe bacteria that are shaped like spirals? Good, so spirilla. Sometimes you might also hear the word spirochete. Okay, usually spirilla or spirochete bacteria are not our best friends, but it varies depending on the species. All right, number 6, what's the capsid on a bacterial cell? What's the capsid on a bacterial cell?
It's a shield to protect and prevent harm from the bacteria.
Good. It's this outer covering on bacteria that protects them. Number 7, what is the cell wall on a bacterial cell? What is the cell wall on a bacterial cell? So it's made of peptidoglycan. Good. And if we, oh my God, I am ready to just itch my nose off. Okay, -5 Ms. Rosna. So if we go back to the diagram here, the capsid is this peach outer covering. The plasma membrane is this yellow inside layer, and sandwiched between the plasma membrane and the capsid is this brown layer called the cell wall. Okay, so the cell wall is made of something really weird called peptidoglycan. It also helps give the bacteria its very unique shape and structure. All right, number 8 and 9, we talked about at the beginning of class. So gram-positive bacteria have a very thick cell wall. Gram-negative bacteria have a very thin cell wall. Number 10, do bacteria have genetic instructions like our cells do?
Yes.
Yes, right? They are a living thing that reproduces, and living things are able to be living and reproduce because they have genetic information. So while it's not the exact same genetic code that's in our cells, it's still DNA. It's still made of the same things our DNA is made of. Number 11, do bacterial cells complete protein synthesis like our cells do?
Yeah.
Yes. All living things have to do protein synthesis. I know from the diagram that they do because not only do they have DNA, but they also have ribosomes. And if we think back to our last unit, what were ribosomes used for? They were used to make proteins. Proteins, again, are what make all chemical reactions happen. When we talk about processes that are happening inside us or inside the bacteria that keep us living and healthy, these processes are actually very extensive series of chemical reactions, and they're driven by proteins. The shape and the color and the look of everything in our body, but also on the bacteria, is because of proteins. So yes, they produce proteins. Number 12, what do bacteria use their flagella for?
Movement to propel some type of bacteria.
Good. So it's a long whip-like tail. We see it here. It's this gray-blue squiggly line that bacteria can actually whip around to help them kind of propel and move through liquids. So just like we require lots of water, bacteria require some sort of moisture in their environment to stay alive. And we actually have proof in petri dishes and fossil evidence where we can see the little pathways bacteria propel through and actually push themselves through. So we know that they use that and move it. What about these little hair-like structures called pili? What do bacteria use those for?
Used to transfer DNA and for attachment to the surface.
Good. So pili actually have very special functions. One, they can help bacteria grip to surfaces, which can be good for the bacteria, bad for us if it's infectious bacteria. But what bacteria can also do is copy and share their DNA. So let's say this bacteria here had a certain genetic trait that was really good and awesome and helped it do better and survive. This bacteria can actually make a copy of it and then share it through these little hair-like structures to other bacteria. So think about it. Think that means for any of you in this class right now where school just always kind of came easy, you put a little bit of effort in, but not too much. Maybe part of that is part of your DNA or genetics. What if we could make a copy of that part of your DNA that allows you to retain information easily and connect it to learn it, and we could share it with all your other classmates? That'd be fabulous, right? Well, bacteria can actually do something like that, okay? And that's actually going to play a very important role in understanding Addie's case. All right, we're going to move on to antibiotics. Okay, so it starts here by talking about number 14. What are vaccines and how do they work? What are vaccines and how do they work?
Um, a type of service that gives someone or something immunity from particular diseases.
Hey, good. So it's what allows someone to be immune to different types of diseases. So immune meaning that our immune system or body is able to prevent ourselves from being infected by it. So there are vaccines for bacterial infections and there are vaccines for viral infections. Okay, a little bit different than antibiotics. A lot of times vaccines actually include small amounts of the structural parts of the bacteria or virus in it. Not the same as giving you the virus or bacteria, just structural cell parts. And that's because when these foreign parts get in our body, our immune system will then make antibodies against these foreign parts, which eventually long-term protects us from those viruses or bacteria. The newest vaccinations, like for COVID-19, scientists actually use an even smarter concept. So their idea was instead of injecting people with parts of the COVID-19 virus, what if we actually just inject them with some messenger RNA that tells their body to make the necessary antibodies to protect them from COVID-19? So they used our understanding of DNA and RNA and protein synthesis to make a new, different high-tech vaccination. Number 15, what are antibiotics and what is their function? What are antibiotics and what is their function?
It's used to fight infections caused by bacteria.
Okay, and when we say fight infection, what do they do to infections like bacteria? What do antibiotics do to bacteria? So when we say it fights them, what do we mean by that?
is it like it's killing the?
Yeah, that's the bacteria. Okay. Number 16, the 3 types of antibiotics are the ones listed below: beta-lactam, macrolide, and quinolone antibiotics. What do beta-lactam antibiotics do to bacteria? Do beta-lactam antibiotics do to bacteria? Very good. So Kendrea messaged me and said it blocks the ability of bacteria to make cell walls. Awesome. So cell wall is like the outer covering of the bacteria, like your skin is your outer covering. So that means if there's ever any damage to their cell wall, they can't repair it. They will die in the presence of beta-lactam. If they're trying to reproduce, they can't, because part of reproducing is making a new cell wall around the new bacteria, and they can't do that, so they can't reproduce. So beta-lactam antibiotics don't necessarily kill all of the already existing bacteria, but they do prevent those bacteria from reproducing, which means it allows your immune system to just focus on one non-growing population of bacteria to kill. All right, number 18, what do macrolide antibiotics do to cells? What do macrolide antibiotics do to the bacteria?
They block bacteria's protein building.
Yeah, they prevent the bacteria from making proteins because it actually interrupts the ribosomes. So again, those proteins that do everything in the cell, run all the chemical reactions that keep them living, that make all the structures of the bacterial cell, no longer are being made. So in the presence of macrolide, bacterial cells very quickly die. Macrolide antibiotics are a little bit stronger than beta-lactam, so they'd be used in a little bit more resistant and a little bit more widespread bacteria. So if we have a little bit of a bigger infection, we might use a macrolide. What about a quinolone antibiotic? What do those do? What do quinolone antibiotics do?
When bacteria begin their protein synthesis process, the strands break off.
Yeah, quinolones actually destroy bacteria's DNA. Quinolone antibiotics were like, screw all this like messing with these structures in bacteria. Go to the heart of it. Go to where the cell gets all of its directions to function and exist. DNA. Quinolone antibiotics destroy the DNA in bacterial cells, and that's how it kills them. So they're usually the stronger class of antibiotics. Okay. Alrighty. So we went over your homework. We're going to update our evidence tracker, just remind you what it is and everything that's there. Again, in your evidence tracker, I am keeping record of everything we learn, everything we observe and figure out. So that way you have one place to kind of access all of this useful information. Okay. So again, we started with looking at Addie and her video. We looked at the CDC poster, a methicillin graph. I feel like every time I say meth graph, people think I'm talking about a graph about drugs, but I'm not. We looked at where we can find more bacteria. We learned what MRSA is. That's one of the types of bacteria that Addie had inside of her. We learned that antibiotics kill bacteria, and if bacteria are resistant, that means they can't be killed by the antibiotics. We know that bacteria make more of themselves very quickly, and they do it through a process called binary fission. We then calculated and graphed just how fast a population or group of bacteria can reproduce and increase in just a matter of a few hours. And then today we looked at the different types of bacteria, what made them different, what bacteria are made of, and types of antibiotics. Okay, so this helps us look at, are all bacteria the same? What makes them different? What makes different antibiotics different? Okay. Again, you have access to this within the modules, but it's also a button on the home page of the course that you can always access from there. All right, so we are ending class today looking at your homework assignment. This homework assignment will be due tomorrow night, and you are going to create a bacterial model. Now, we've made lots of models before. I'm going to start by going over what your model needs to show and answer. I'm then going to review what a model is and give you some tips on how to start, okay? So this model needs to show and answer what are bacteria, what are they made of, what are the different types, and how do they reproduce? Okay, all information we've gone over. Now remember, when we are modeling, that we want our model to be mostly visual. So if I look at this example of a model here, notice I don't have like long sentences or paragraphs of text with then like some random little icon above it, like one bacterial cell or a stick figure to represent like the visual part of it. It's almost like I were to pause an animation or a video showing a process and take a picture of that. Okay, that's what this model is like. I can physically see in the model processes happening, different structures and parts involved. I use different colors to represent different substances, different types of labels. I can use arrows to show that something is moving. I can use different shapes. I could zoom into a picture to look more closely on the inside or more closely in general. I could zoom out to look at a bigger picture. Everything is labeled so that no matter how good of a drawer I think I am, if I clearly labeled it, people know exactly what I'm showing. Okay, and again, mostly visual. So I know the number one question I get is, how do I start this? Or I don't know even how to begin or start this. So if I were making a model, the first thing I would do on a scrap sheet of paper is list out my answers to these 4 questions. That would be step 1. Now that I have them all answered, I know exactly what I need to show. So then on another piece of paper, I would start brainstorming or sketching out, how can I take this information or these sentences I wrote and draw them or show them visually? Okay, then I would start to draw that. I'd make sure, again, I label everything. But again, I would first start by answering these questions. And once I know exactly the content and information that I have to show, now I can begin brainstorming, how can I show that, draw that, or represent it, okay? You are able to hand draw this, which I think most of us usually choose. You can then just take a picture of your model and upload it to this page. I will first give some tips on that. If you are going to hand draw it, please do not do it all in pencil. Graphite or pencil is very reflective. So a lot of times when you take pictures, light bounces off your pencil and it's difficult to see. Some of us write very lightly, which makes it more difficult if it's pencil. So I would recommend like blue or black pen, or if you're using markers, that they are bold colors, not like highlighters, for instance. When you take a picture, make sure you're in a brightly lit room and that you're taking a picture directly above your drawing. Okay, so don't try to take it at some weird angle, in a dark corner of your bedroom, things like that. Okay. This model, once again, is due tomorrow night. You have the remainder of class today to work on this. So this actually gives you about 40 minutes to start it. And for some of you, that might be enough to finish it. Okay. Does anyone have any questions about what you have to do in this model, when it's due, or anything like that? Then you have the remainder of class time to get started working on this. Again, if you ever have any questions, drop them in the chat or email me, whatever is available and comfortable for you. And I am done bleeding for today.